Chusoy from our own Department of Chemistry. He came all the way over from Weiser Patent today. And if anybody would like to escort him back when it's over, help him find his way, I'd appreciate it. Charles, he grew up in McLean, Virginia, which makes us practically cousins because I'm from Virginia too. And he was also educated in Virginia. He went to George Mason University for his bachelor's degree. James Madison. Yeah, universities named after presidents, I know. So, or early patriots. So he went to James Madison University for his bachelor's degree, and then George Mason for his master's and his PhD. And after that, he did a postdoc at Texas A&M in conjunction with, what was it? It was Pacific Northwest National Lab. So he was in 2 places at once. I don't know if you can tell us how you do that. But I did that one. And after they took his first position was at Missouri University of Science and Technology, and he stayed there for a few years before we had the pleasure of getting to know him. So he's been here since 2010. So Charles,
thank
welcome.
you. Yeah, to answer your question, I basically was fully funded at the Pacific Northwest National Lab, but I did all my work at Texas A&M, and I just had to fly back to Washington to tell them what I did, so that's how I did that. Anyway, thank you for coming to my seminar. Actually, the motivation for my presenting of this seminar, because I know some people know that I've been doing work on carbon nanotubes, but basically I had previously planned to teach this course in the fall, surface characterization nanomaterials. So I was thinking of a topic that would appeal to a wider audience and basically base it on the work that I've done, previously, back in Texas, and so hence the topic of my talk for today. But unfortunately, I didn't understand the scheduling of courses, so it looks like I'm not going to be teaching it this fall. You'll have to wait a year. So for those of you who are thinking about elective courses to sign for, it'll be in 2013, not 2010. So anyway, so that's the late breaking news about that. What I want to talk about today is some work that I've done on characterizing bone growth on model artificial implant surfaces. And basically, the 2 surfaces that I worked on primarily is like the titania surface, TiO2, and also looked at self-assembled monolayers that were functionalized with carboxylic acid groups. And so this is work that I've done way back when I was working in Texas and at Pacific Northwest National Lab. Now, the motivation for this work comes from the fact that when people get like artificial hip replacements or they tend to do it kind of late in life, because the reason for that is because we don't know how to make the adhesion between living tissue, from the bone and the artificial implant itself last for a long time. Basically, maybe 15 years, 20 years if you're optimistic or so. And actually, the way we do it today is, we bolt things on and hammer things together, and hopefully it'll hold. So basically, that's why people don't get hip replacements or knee replacements or artificial hip until they're later in life, because you don't want to get that done again. So if you're going to have an operation, just have it done once. So the challenge to basically be able to improve this type of adhesion is to first understand and develop our ability to characterize the phases of calcium phosphate that can form. So calcium phosphate exists in a variety of forms, variety of structures. Octocalcium phosphate, this is dicalcium phosphate dihydrate here. This is an amorphous phase. There's different phases of tricalcium phosphate, one's monoclinic, one's rhombohedral. And so all of these, it can basically, grow and nucleate and grow. And the type of, and the phase that's prevalent in bone and teeth is the hydroxyapatite. And so that's the most thermodynamically stable phase, and also that's the strongest phase that we have. Now as far as growth of this type of material onto surfaces basically occurs in 4 steps. One is that there's a modification of the surface material due to chemical interactions or cell activity, followed by nucleation of carbonate apatite crystals from, let's say, body fluids, and then followed by protein absorption and osteoclastic adhesion and expression fibers. So that basically just forms the bonding between like the living tissue, which is the bone, and then the implant itself. Now, as far as the scope of my talk, I can only address steps 1 and have a chance of addressing step 2. Steps 3 and 4 are basically beyond the scope of my study, but at least I can give you an idea as far as how we are able to at least identify the phases that are present as the material forms, and so which is an important first step. So actually, I have this in my laboratory over at Weiser. Basically, this is a constant composition apparatus. What we can do here is basically this apparatus is consists of a solution pH. You're going to have a magnetic stir, and then we can have, let's say, model metal oxide surfaces in solution. That's basically tied to a titration unit that basically can feed in, let's say, phosphate, calcium, etc. And this could be, dynamic. So basically, this mimics like physiological conditions. So when basically material grows onto these surfaces, and for the first part of my talk, I'm just going to talk about metal oxides. There's going to be a depletion of those ions in solution, and so when that depletion occurs, basically this titration unit basically pumps back that in just to maintain the same conditions. And so, if we do that, basically we looked at three different metal oxides: TiO2, aluminum oxide, and silica oxide. Basically, these are just other oxides that we used for comparison. And it turns out that if we monitor the amount of moles of calcium phosphate by ion selective electrode, we find that, let's say, the depletion of the phosphate in solution is correlated with the uptake of it onto the colloidal seeds. And so these are all normalized to the same BET surface area. We find that the calcium phosphate grows fastest on the TiO2 rather than on the aluminum and the silica surface. And that's attributed to the surface structure of the TiO2, where basically the phosphate groups can link to the hollow sites more effectively because of the geometry. And so that was done using that data was obtained using this constant composition apparatus. Now one question is, okay, now how do we identify or how do we characterize the form of the phosphate? And so this is where this machine comes in. Okay, and I have this in Weiser as well. And so basically this is an X-ray photo electron spectrometer. It also has a mass spec into it. So basically this is a sample probe. It's kind of home built here. So what we can do is take the sample. Basically, we can prepare planar model metal oxide surfaces to simplify the structure. So we can basically make some characterizations on the onset of the calcium phosphate growth, just as it occurs. And so basically, we do this by it turns out that in order to do electron spectroscopy, you have to overcome something called charging effects. And so we do this by depositing thin film surface of TiO2 onto the substrate here. So that's about 15 angstroms thick. It turns out that that thickness is sufficient to have all the chemical properties of the TiO2, but also you can overcome charging effects because it's thin enough. Because usually metal oxides, they're insulators, and that's why you have problems with charging and getting full resolution and spectrum. So basically, we do this by inside this vacuum chamber. There's a little doser there, and we basically have some titanium wire here that we basically attach to this tungsten filament, and then we pass a current through it, and then the metals from the titanium metal will basically combine with oxygen oxygen needed about 10 to the minus 6 torr pressure. So by the way, the pressure inside here is about 10 to the minus 9, 10 to the minus 10 torr. So it's low pressure research. So anyway, so that's how we form the surface. And of course, the advantage of this is that we know it's clean. We know it's only TiO2. There's no interferences with atmospheric gases that are absorbing onto the surface. And we can basically take this, and then we have a home-built a liquid cell. Okay, I also have that in the lines. Actually, I got 2 of them. And when we put this in this chamber, so that's where the liquid cell goes in. So basically, you can take this sample, put down TiO2 film, thin film, a clean, atomically clean film, and vacuum. Take it out here, and then react it with the same conditions as here, and then bring it back into the system, and then surface analyze it. So that's what we've done here. So basically, we deposit fresh TiO2 overlayers, and then we did this by passing a current through it in, let's say, about 10 to the minus 6 torr oxygen. And then we basically reacted it with these solution conditions, which were found to cause nucleation and growth of calcium phosphate onto the fluorescence. Okay. And so this sample probe can be rotated and inverted, and so this is what the sample probe looks like. So this is basically a substrate where we basically evaporate the TiO2 on. This is a thermocouple that's attached to it. So this is a top view, this is a side view. And one question that you might ask is, okay, well, wait a minute, you're doing these experiments in ultra-high vacuum. Aren't you interrupting equilibrium conditions? Well, it turns out that it's okay. Basically, we're able to get complementary kinetic data. And to explain this data, I have to explain this technique here. It's called temperature programmed desorption. Basically, we have a mass spec here. That's a quadruple mass spec tied to a unit. And this mass spec, it's a type of mass spec where you basically tune the mass to charge ratio to a certain value. In this case, 79 for the phosphate. And basically record the signal, and then basically you can move that signal, take the integrated peak area, and basically do a quantitation of the amount of phosphate that absorbs on the surface. So this is the in situ data that we saw from the constant composition apparatus. So if we go down here, this is using the ultra-high vacuum data. So this data here is replotted right here, and this data here is taken from the mass spec experiment, series of mass spec experiments. And so all you can see is that there is, you can see that there is a correlation between at least kinetic growth of phosphate between both the ex situ and in situ data. Now, of course, the caveat is, yes, you are interrupting equilibrium, but it looks like roughly, you can match the behavior. So one question is, okay, so since we've got all these phosphates, and basically when bone grows, there's a transformation of one phase to another. How do we track that? How do we figure that out? So basically what we did is we figured a way to identify those phases. Now, one obvious way of doing it is doing X-ray diffraction. Of course, you can have these characteristic peaks from the X-ray diffraction pattern, but what happens if you have a very small amount of material in it? So let's say we have, 15 micromoles per square meter of calcium phosphate. That's basically at the onset of nucleation and growth. How do we analyze things that small to be able to characterize the phase? Well, we basically just used a conventional XRD. I mean, you can see that there are no peaks that you can see from that. And that's after we took the material and interacted with the solution where we think the onset of nucleation and growth would occur. There's no signal there. So, so the way to do that is to do electron spectroscopy. And so inside that chamber, we see the X-ray gun, what happens inside there. Well, XPS is basically a technique that is based upon the photoelectric effect. You shine X-rays here, it basically knocks off a valence shell electron, and basically there is no damage to the nucleus of the atom. You're only sampling the outer core of electrons. So you can do non-destructive elemental analysis. You can also do chemical oxidation state identification. That's what we will rely on to identify the different calcium phosphate phases. And so this is what the XPS data looks like. This is after, let's say, the TiO2 was exposed to solution after 10 minutes and 12 hours. So this is the oxygen from the TiO2. This is basically oxygen from material that's absorbed onto the surface. So you can see that there is a growth of this oxidation state. This is after 12 hours. So we can monitor the kinetics of growth by looking at the integrated peak areas of the electron spectrum. Now, the binding energy, so there's like specific binding energy positions that we can use as a way to like fingerprint the different phases. Now right here, these correspond to the binding energy peak centers of the different spectra for phosphorus, calcium, and oxygen. And so what I have here, this represents here an error bar. So basically did multiple measurements, but you can see that with the error bars here, I mean, there is a way to distinguish between, let's say, hydroxyapatite, tricalcium phosphate here, but there is quite a bit of overlap. Okay? So basically, XPS wasn't the only thing that we used. We also used secondary ion mass spectrometry. And so this technique basically is based upon bombarding the surface with primary projectiles that results in the release of secondary ions and neutrals, which can be detected. And then again, that could be used as a way of fingerprinting the type of phase that forms. And basically, the physics of SIM, this is secondary ion mass spectrometry, isn't well understood, but we can still use it analytically. And what I want to show here is that we basically use different projectiles, bombard different projectiles on the surface. So it turns out there's signature peaks that we find, basically phosphite to hypophosphite ratio. That was the key to allowing us to identify different phases. So right here, I wanted to show, this is a computer simulation that I pulled off of Professor Barbara Garrison's website at Penn State University. Sorry, that's the wrong. So what I'm showing here is a gallium atom here that's used as the projectile. But notice here, it bombards and impinges the surface. Look at all of this surface damage right here. But there's little release of secondary ions that we can detect. Okay. So this is a top view. And you can see that the surface is disturbed, but basically very little of the secondary ions is produced in that fashion. Now, if we did the same thing, but let's say looked at a heavier atom, let's say we used Buckminster fullerenes. It's a heavier projectile, but interestingly, it causes less surface damage and maximum secondary ion use. So you see this. There's a lot of secondary ion use, but if you look at the lowermost surface, I mean, this remains relatively undamaged as compared to the gallium. And so, basically, this is the top view here. You can see the secondary ions that emerge from that substrate. So the trick is, to optimize the type of analysis, we basically had to bombard the surface with different projectiles to get the optimum, let's say, phosphite to hypophosphite ratio. And let me explain what I mean by that. So we were able to do this by, this is a home-built setup at Dr. Emil Schweikert's lab over at Texas A&M, borrowed his machine. This setup is basically this is a mylar foil that can be coated with different cesium iodide projectiles, basically just painted on. It's just painted on. And you could put in buckyballs, you could put in different cesium iodide clusters right there. This is a californium radioactive source, and one thing that's very interesting about californium, when it radioactively decays and emits fission fragments, 180 degrees opposite of each other. So when a fission fragment hits this spark detector and hits this mylar foil, it hits it simultaneously. And so that gives us a way to, with timing, with it, like this starts a clock, this stops a clock. This is a time of flight tube. But basically, secondary ions will fly down this tube, which does the mass separation. And then basically, because of this timing here, we can basically do sim spectra from multiple projectiles simultaneously. They can be computationally separated out. And so all these spectra were obtained simultaneously, which just saved us a lot of time. It turned out that this first cluster, cesium iodide, cesium, turned out to be the optimum projectile to use because it resulted in the greatest difference between the phosphite and hypophosphite ratios. So these secondary ions were formed from the calcium phosphate clusters. And we can use it as a way to identify the different calcium phosphates. So this is off the calcium phosphate here. You can see that the phosphite to hypophosphite ratio is approximately 1:1. Brushite here is certainly larger than this. And this is a bar graph plotting the phosphite to hypophosphite ratio. So from this, we can identify, we can distinguish between brushite, calcium phosphate. You'll have to forgive the coloring here. This is the alpha tricalcium phosphate, beta tricalcium phosphate. That's the monoclinic long-behavior structures. This hydroxyapatite here, and just by coincidentally, it turns out you can see that the phosphite to hypophosphite ratio seems to match the thermodynamic stability of the different phases. And that's something that we just discovered. There's an interesting coincidence where brushite here is the least thermodynamically stable phase. You see a greater phosphite to hypophosphite ratio, and then as the ease of nucleation, this gets more difficult to nucleate and grow because it has the most thermodynamic stable phase as a hydroxyapatite. That's the bone and teeth. And so basically we see an interesting trend right there. And now, obviously there's some overlap here between hydroxyapatite, let's say alpha tricalcium phosphate, but if we use this in combination with, with the XPS here, there's enough of a difference in the binding energy positions that we can distinguish between these two. We use both techniques together. And so this is the secondary ion yield when you expose the TiO2 to the phosphate solution. And then this is just the phosphite, this is the hypophosphite. These green numbers, they're basically the ratios. So it looks like that the phase that is still amorphous, but it's closest to the brushite phase of the different phases that we analyze, surface analyze, and that's the most thermodynamically unstable phase. That's something that's not surprising. So, so basically that was done in that. The second thing that we did was we did what's known as a sputter depth profile. So basically we bombarded the surface by bombarding it with a stream of, let's say, argon ions, etched away the surface, and did electron spectroscopy now. So this is being argon. I've got that set up in Wiser as well. So and then we can basically do a compositional profile as we etch away the surface. So this is what our data looks like. This is an XPS uptake of calcium phosphate on the TiO2. So we can see phosphate grow here. You can see the calcium grow. And so this corresponds to 1 monomer equivalent. So basically, we just took the sample phosphate, assuming there's 10 to the 15th sites per square centimeter spin coated on the surface, and then did an XPS. I mean, that's what that horizontal line is. And this is basically corresponds to that small amount of material where we think nucleation rate begins to occur. Now this is the sputter depth profile. This is when we basically are etching away the topmost layers and then simultaneously doing electron spectroscopy at the same time. What we see here is that we see that there's the phosphorus here, signal here, is greatest here, and as we etch away the surface, we can see that this drops down here, like so. And then at the same time, you see the emergence of a calcium signal here. So like the overall picture is that it looks like the initial layer as the material grows, it's calcium rich, and then the phosphate groups grow on top of that, and so we have evidence for, let's say, phosphate island, and hence the cartoon right here. So this is the TiO2 surface, calcium, and then a phosphate island, and so we're etching away. We're basically blasting off the phosphate here, so that's why the phosphorus grows down here. And this is the structure that we basically etch away that much material. So, like the overall picture in terms of absorption on TiO2 is basically evidence for island. Now, one question that you may ask is, you can't have negatively charged groups that just grows on the surface. What's the charge balance? And I mean, that's a good question. Like, if it's not calcium, what is it? And the only thing we can think of as far as postulating is like the hydronium ion that may serve as the charge balance. So, but anyway, that's a very interesting question, and I know this isn't, I don't know, may or may not be convincing, but that's the best explanation that we can think of. And what we see here, this is basically a schematic that kind of explains this structure. This is an atomic force microscope image where we can, after a 90-hour exposure, you can see some evidence for islanding on the TiO2 surface. And the way I've drawn this scheme here is that you can see that this is like a brushite-like phase, where basically we have like hydrogen serving as the charge balance. And unfortunately, XPS, you can't do analysis of the hydrogens. That's a limitation of the technique. That's what we have. So, in conclusion, for this first part, we see that as calcium phosphate grows onto TiO2 metal oxide surface, we see that it starts by an initial calcium-rich layer, followed by phosphate island, and then we just postulate that this is the charge balance. This is the source of the charge balance, and so we see brushite-like characteristics, and that's the least thermodynamically stable phase of the calcium phosphate. So we don't see hydroxyapatite immediately form, but this is a very early stage of nucleation growth. It turns out that in the next part of this talk, you see that it requires much longer exposure times to form that phase that we find in both. And then another thing is that in doing this scheme, I basically borrowed some ideas from intersphere complexation, that geochemists or geologists use, where basically materials bond onto those sites of, let's say, a mineral oxide. So that's where this scheme comes from. Okay, so that's part one on the mineral oxide. Part two has to do with looking at interactions with some organic groups, and basically, so we just kind of, for the metal oxide, we basically looked at this, but at least with this second part, we can have an idea of taking a look at nucleation growth of apatite crystal from body fluids. So that's where self-assembled monolayers come in, and I'll explain what that is in a moment. Again, we can have various phases that can coexist, and we want to be able to control phase as things nucleate. So again, I think in the first part, we addressed the challenge of, let's say, how to characterize those phases. Now, with self-assembled monolayers is basically a way to create very ordered organic structures on a surface. So usually, we take a piece of gold or gold that's been sputter deposited on, let's say, silica, like silica 111, and then you get a gold surface. That's a cheap way to do it. Instead of getting gold itself, at least just have it sputter coated. And then we can use immerse it in an alkane thiol solution. Basically, it's just an organic chain with a sulfur here, lots of carbons, and then you can have your functional group of your choice. And so what was discovered, by the group of like David Alera and Ralph Muzo back in Penn State University back in the 1980s or so, was that if you did this, you'll find that those molecules will self-assemble in an ordered manner, so that the sulfur attaches fairly well to the gold surface. And then the head groups, they tend to be, these are very well known as far as the positions. So they're like, 4.97 angstroms apart. We know it's very ordered, and we can put down end groups of our choice. And, so these make very nice surfaces to do very well-defined cells. And so this is our carboxylic acid SAM. So what we find is that carboxylic acid groups appear periodically, let's say butanic acid, like in these sequences, that are in the collagen, that in the involved in formation of bone, attached bone to bone tissue. Let's see. I don't remember the sequences. Let's see. Maybe I got. Oh yeah, right here. No, I guess I. Oh yeah, there are residues at. This is 61, 78, 130, 61, 51 sialoprotein, and so those are where the carboxylic acid groups show up in terms of it turns out that we have a way of this is basically taken from a landmark study by George Whitesides and Joanna Eisenberg. Basically, not only is it possible to form these groups, you can basically make a PBMS stamp, like use an inkjet printer to pattern a surface. So all of these, let's say, so this is, see these little stars here, it's hard to see, these are carboxylic acid terminal groups. The rest of these are methyl terminated groups. Okay, so this is some microscopy of this, and then what they did is, what was found is that calcite, biomineralization preferentially grows onto the carboxylic acid groups, whereas there's no growth on the methyl groups. So basically, the functional groups matter in terms of bonding, with the oxyanion. In this case, in the case that we're talking right now, phosphate, the oxyanion of calcium phosphate. Okay. So in case, so nucleation and growth are mediated by an organic interface here, where we have these carboxylic acid groups at these residues, and so that's why we focused on the carboxylic acid. And then one thing we wanted to find out was what is the mechanism of this nucleation and growth. There's some theoretical patterns here. This is the delta G that corresponds to what makes this nucleation and growth spontaneous. And that's what we've sort of investigated here. So what we did is we took the self-assembled monolayers and then immersed it in the solutions that mimic physiological conditions here. So this favors the growth of the different calcium phosphate phases. These are the concentrations that were used. This sigma here is basically parameters that were adjusted such that certain phases will grow, will appear. And so, of course, that was determined, theoretically based on this model. So if we adjust the solution parameters here, we can basically try to grow these different phases of the calcium phosphate. And again, we relied on our surface analysis techniques. Another thing that we did was, we, in terms of monitoring the nucleation growth of material, is we used a quartz crystal microbalance. So basically, this monitors, this monitors the change in the mass. The change in the mass corresponds to the growth of these materials on that surface. So quartz crystal microbalance, we can monitor change in the nanogram regime, and we also couple this with our electron spectroscopic analysis and SIMS analysis as well. So factors affecting nucleation and growth. For one thing, time. Okay, that's first thing, time, and functional groups here. So we did these experiments with carboxylic acid, functionalized SAMs, hydroxyl functionalized SAMs, methyl functionalized SAMs, and we can see that growth occurs the fastest on the carboxylic acid because of the attachment of the oxyanion to the base. And so this is measured by using the quartz crystal microbalance. Another thing that affects the rate of growth is the concentration of the calcium. Okay, so if you vary the calcium here, so you can see a greater concentration of calcium here results in faster growth. Temperature also affects this. So this is growth on the carboxylic acid functionalized SAMs. This is at room temperature. You see slow growth, and then at physiological temperature, it grows faster. So in terms of trying to form the crystallites, we use the physiological conditions at pH 7.4 and also this physiological temperature as well. One thing that you might ask as far as some of these surface handling techniques, you might destroy some of the surface during the analysis. So this is a SIM spectra of the carboxylic acid functionalized SAMs after interaction with phosphate. And this is after this has been photooxidized. So basically, one way to actually destroy the material is basically take a UV lamp and shine on the surface for about 25 minutes or so. That basically causes it to, causes the SAMs to be decomposed. And so that's why you see the SO4, SO3. Now it turns out that after the SIMS process, we see no SO4, SO3. So we can be pretty safe that we're not damaging the thing that we're trying to analyze in terms of phase characterization. And we also rely on XPS core sheets. Now this is the kinetic uptake. Okay, so we see at the very early growth here. Again, you can see that in the previous study, we absorbed it on for a fairly short period of time, to 5 hours. What we see is that this is growth on the carboxylic acid SAMs, OH and CH3. Nothing happens here, but we can see that there's a fairly rapid growth, and then it tends to remain stabilized. So there's fairly weak absorption. There's some absorption of some material, but we see no crystals yet. Bone has not yet formed. It turns out that we needed to do this reaction for a longer period of time. So we actually saw initial. The mass started to change rapidly after about 200 some hours, or almost 300 hours, basically 12 days. And so this is with the data from the quartz crystal microbalance. So all of these are data points from the quartz crystal microbalance, and this is data from the solution composition from the constant composition apparatus. So we can see that there's a very good correlation between the mass, change in mass, and also the ions, where depletion of ions corresponds to growth on that surface. Okay. So these are data from our time of flight SIMS. Again, this is noting the PO3 to PO2 ratio, phosphite to hypophosphite. And then this is at the early stages of growth. But even at these low stages of growth, we see evidence for, let's say, formation of octocalcium phosphate and hydroxyapatite. So if you just look at this, we can actually see the phase formation onto the carboxylic acid SIMS. And so that basically corresponds with what we see here and here. However, even for a short period of time, we really don't see any crystallites. So, but then again, we're looking at a very small amount of material. We're looking at, let's say, 50 to 80 angstroms thick material. So we need to put it in the solution for longer periods. So this is what the SAMs looks like. So these correspond to groups. These are terraces on the gold substrate. So it's just to give you an idea of what the flat surface looks like before we put on the calcium phosphate. These are pits that are present on the SAM surface. I guess you can't get away this is a close-up at 200 nanometers here. If you zoom out, and these are just terraces that exist on the surface here. You zoom out, and this is after 13 hours of the growth. This, 353 hours. So growth started around 340 hours, where we begin to see some of the crystallites that begin to form. So this is an enlarged view. This is a 45-degree tilt view. Okay. And then after 358 hours, you can see that once when you hit like an induction period, things start to rapidly grow. And so you see 18 hours of growth. You see things fill in. I mean, obviously, it doesn't grow beyond that initial first layer, but you can see how it fills in, and then this is after 53 hours within the growth, 398 hours here, and so this is zooming back out. We can notice 3 microns, 5 microns, 3 microns. So we can see that after an induction period, this seems to rapidly grow. And that's what close-up of one of these structures looks like. And so isolate crystallites appear on the surface, crystallites grow and coalesce, and then tend to grow vertically. And then you look at the glancing angle XRD, you see that there's a lot of structure here in the 002, orientation, which means that you have plate-like substructures that are perpendicular to the surface, to the same surface. All right. Now, to kind of explain, what's happening here, it looks like at the early stages of growth, looks like you have some absorption from the calcium and phosphate, but then something else needs to happen, and it turns out that it would seem that this model is consistent with what we think is happening. Is that there is a nucleation, like a nucleation occurs in the solution itself. And after it's basically a critical cluster nucleation mechanism. So nucleation has to occur in solution, where basically it needs to reach a critical point, and then afterwards it deposits onto the surface. And so that's where we see the rapid growth of the calcium phosphate. And this is also supported by, if you basically took a laser pointer and then shined it into the solution, you would see a scattering of light from the Tyndall effect, meaning that there's a formation of those critical clusters prior to deposition on the same surface. So at least experimental evidence is consistent with this model, this critical cluster nucleation mechanism. So to summarize these findings, we find that the calcium phosphate film formation occurs most rapidly on the carboxylic acid sands. Of course, that's consistent with what's observed for, let's say, other types of oxyanions interacting with carboxylic acids in the literature, increasing the calcium ion concentration accelerates the surface deposition here, and then long induction periods are needed before we see those crystallites begin to form. Okay, and then and so we see images of these crystallites coalescing on the sands after that induction period. And then the XRDs seem to indicate that they're plate-like structures, also complementing the images, the electron microscopy images. And there's a high degree of orientation, and then just before we see a hydroxyapatite, we see octocalcium phosphate first. And actually, that's consistent with what we know about Oswald's step rule. So, anyway, to acknowledge the funding. Oops, excuse the technical difficulty here. It's not letting me. want to acknowledge the funding. We had funding from NSF to do this work. Also, I want to acknowledge my collaborators over at Texas A&M. Okay, I got it.
It's fine. It's fine. We can read it.
Good job. All right, okay, I'm sorry. My apologies. My apologies. I didn't think this would happen. I don't know what happened. Okay, so I want to acknowledge the Associated Western Universities for sponsoring a postdoctoral fellowship that supported this work, an NSF grant here, and then of course these are our postdoc, Michael Van Stipdok. He's now a professor at Wichita State University. His graduate students, Neil Schleicher for the SIMS apparatus that they let us use, and for Wayne Goodman, my advisor, who let me buy that piece of instrumentation dirt cheap, so that I have advisor right now. And then also these folks who are involved. These kind of got me interested in biomineralization stuff. Anyway, thank you for your attention. I think I'm on time, so and I'll be happy to answer questions. Yes.
What is the hydrochloric acid secreted from osteoclasts to interfere with binding calcium phosphate to oxalic acid?
Well, I know the solution conditions were adjusted to pH 74. So that was controlled. Acidity was controlled.
Okay, I see.
Yes,
I'm wondering about if there's a clear view of the downstream applications of this. Because you grow the crystals, then what is there a plan for a little too far in the future for what you do with the crystals?
Well, I don't know. Of course, we're just looking at like the very early stages for, let's say, osteogenesis, etc. After that point, I think there's some biology that needs to happen that I don't understand. That's beyond my area of expertise. But as far as nucleation and growth goes, it only grows up to like a certain vertical height. It doesn't do that indefinitely. Just like once when the first monolayer is achieved, that's it. So I mean that's as far as what happens chemically speaking. I think the biology needs to come into play after that.
So you can coat the pins or whatever that you do.
Yes. Well, now some practical applications of it. There's certain there's certain devices, implants that people use, let's say heart valves and things like that. Now certainly, if you want to avoid the formation of crystals onto like heart valves that would basically stop them from functioning, I mean, you definitely want to have certain functional groups, terminated with certain functional groups. Like you would want to have like the carboxylic acid, for instance. So I mean, that's one application. One thing that you can understand is like the surface. The way the surface is terminated influences how crystallites grow. Yes?
I was reading just a couple of weeks ago, there's been a real rash of implants that have been failing 10 years early. And this is a new kind of material they've been using only in the last 5 to 10 years. And I think it's some kind of, they used to use titanium, I'm not sure, but they changed materials. This new implant that you're using about 10 years, and they expect a 20-year lifespan, but they're finding that they're getting nowhere near that. Does anybody know what material been on the news last couple of weeks? If it's related to material, is that anything relevant to what you're studying?
No, I'm afraid I'm not familiar with that new material.
Yeah, a lot of people got hip implants and everything like 5 years ago. It's been terrible. They can't walk anymore. It's like 30–40% of it. It's huge.
I think
Huge
that's,
failure rate.
I
I'm not mistaken, Kristen, that is partly a failure of the implant onto the bone.
Yeah, which materials
the
was
bone
it?
isn't
Yeah.
growing up
It
around the
wasn't,
implant
yeah.
properly, so it's not holding, and then it will break off.
Yeah.
And then it actually, now you've got a bigger problem because now you've broken off a bigger part of the
Yeah.
bone in addition to
Yeah.
loss
It's a
of
problem with the
function.
new material, but it wasn't a problem with the old material. So I'm wondering if, do you remember what materials were in the new implants?
I think it's a combination of the materials shift and they thought that this material would work better with bone, so they didn't set screws as deep. Yeah, they used to not rely on the bone growth on the material at all. And then this new generation, they thought they would get bone growth on it and they didn't.
Yeah.
So now you're getting failures much earlier with the aging population that gets the implants.
Yeah, well, the news is certainly relevant to getting funding for this
Yeah.
research.
Certainly. Yeah, I can't comment, I don't know enough to comment on
But
it.
it's important research and development.
That's certainly, that's certainly an area for funding, and obviously it's something, you know, if we get funding, then we'll do research on it, so.
I think that's
Money,
where-
I'll work on it, so.
I think that's where technology comes in, I think it's one of those deals where they originally did some work in vivo, or excuse me, in vitro, and had bone growth on it. And then when they went to the live models, when they went to people, it didn't.
One thing I can just comment, is that it's the usefulness of a surface science approach. That is, looking at materials where the structures are well defined.